I think you should be able to see my slides correctly now. But yeah, so I think just to start it off, I just have one slide to we're going to mostly be talking about AI and machine. Actually, I'm going to move you guys over here. We're going to mostly be talking about AI and machine learning related stuff for this one. But since this is data science for the non-data scientist, I just wanted to give this little visual that I like to do for like, what is data science? Because I have some friends who say data science isn't real, but it's really just because it's a collection of skills. Yeah, I'll just move on from there. You can look at this at your pleasure, but let's first start a little bit about AI and machine learning in basic science research, because I think that's one. I guess the point is, like, machine learning is being used pretty broadly in the biomedical space and really all sorts of sciences at this point. It's not always obvious like where it's happening, but I'd like to just point out a couple cases where this is coming up. All right, so the first example I'm going to talk about is just in protein folding. And some of you actually are probably aware of this because of some big news that happened last year. Kind of controversial news in a way too. But so in the space of protein folding, if you're unfamiliar, proteins are just chains of amino acids that fold into these complex shapes. And the shape of the protein, the way they fold up, is effectively what defines its function. It has kind of like this lock and key mechanism for describing how proteins work. And so then, because the function of the protein is defined by its shape, knowing the shape can be used for things like drug discovery, understanding disease mechanisms, even in material design, like biology and evolution, and all sorts of other different application areas. But basically, understanding the shape of a protein, like how it folds up, is pretty important. And historically, that's been done through experimental discovery. I believe Chris mentioned that she's an expert, at least on, or somewhat of an expert, at least on one of these methods. It's these experimental processes were kind of slow. Then there were like a few different methods. I know more about X-ray crystallography, but almost nothing about it actually. But there are these other methods as well. But they all have their advantages and disadvantages. But the point is that it always took a fair amount of time to actually get the structure of any single protein using any of these methods. And there were, again, other trade-offs that were involved in those. And then, so that meant that over the last several decades, predicting a protein shape from its amino acid sequence was kind of like this grand challenge, but it was historically really inaccurate, like inaccurate enough that it wasn't like super useful, the methods that were produced. But there were these grand challenges that were happening. And in particular, there was one called this critical assessment of structure prediction. And in 2018 and 2020, I guess their 13th and 14th challenge respectively, there was a model that came out. It was developed by Google DeepMind, or formerly DeepMind, now Google DeepMind. Their model is called AlphaFold. It's a machine learning model, or it's a deep learning model. And it won this CASP 13 and 14 in both in 2020, it won by like a long shot, basically. And it was so accurate that it was almost like as good as some of the other experimental methods. And what was really neat about it was basically, I guess I won't explain, like, I can't explain really how the model works in depth. I'm sure we could spend a whole session talking about that. There's a Nature paper on it, I believe from 2021. But basically, it's this deep learning model that predicts pairwise distances and orientations between the residues, or the amino acids. And how it works is basically it takes the high level of how it works is it takes this amino acid sequence, and here you can see this is myoglobin, which I believe was the first protein whose structure was determined. But it's a relatively short sequence. I think it's 150-ish amino acids long. And so you just give AlphaFold the sequence, and it can tell you a really accurate protein shape just from that, basically. And it works by first looking at homologous sequences, so from the similar proteins from other species, and looking at co-evolving residues that seem to change in the same way at the same time. And that suggests that those residues tend to be close together in 3D space. And then it can optionally use known structures of similar proteins as well. So like if we already have done, like, experimentally determined the structure of some protein that's very similar to that one, that's probably similar in shape as well. And then famously or infamously last year, Demis Hassabis and John Jumper at DeepMind shared the Nobel Prize in Chemistry So in 2024, even despite being computer scientists. So that was, again, for the development of AlphaFold. So that was kind of this big thing that happened last year. So super cool. It's super scalable. Like you can get protein structures from a novel sequence in minutes. And they've already like they've created this database of like, I don't know, like tens of thousands or hundreds of thousands of different protein shapes that have been predicted by AlphaFold. So really neat, can improve scientific process and speed it up. And then and feel free to interrupt me at any time. I'm happy to answer questions to the best of my ability. But just moving along from that, in single cell analysis, in this case, I mean like transcriptomics of single cell analysis. Historically, when we've done transcriptomics, we've thought about like bulk sample, where you have lots of cells together in this one sample, and then you isolate the RNAs from that group of cells, and then you sequence them. But bulk analysis provides like an average expression of a set of cells. So that's what this cartoon on the left is sort of trying to show, is that you got these bluish cells and then this orange and green cell, and they're all in one sample. And bulk analysis gives you kind of the bluish average expression level from that sample. But more recently, and like it's becoming much more common to do single cell analysis, where each of the cells are individually isolated and then sequenced. So you have a sequence for every single cell in some set of cells that you've collected. And the data set can be noisier, but then it does allow you to capture the heterogeneity within some like tissue samples, say, which then allows you to detect, say, rare cell types. In that case, that might be represented by this orange and green cell. You can also then track cell trajectories over time. Like you just get to basically cover the complexity of samples more dramatically, even if the data can be a little bit noisier. Now, that by itself isn't anything that machine learning has touched, but one place where machine learning has sort of changed things in this space is how we do at least some basic initial analysis of some of this data. So traditionally what we would do when we're trying to like determine, say, quality or relationships between samples or cell types is we would do a principal component analysis, which basically just takes the high dimensional data of sequence data, or the expression data, so on the order of tens of thousands of genes, say, that you've measured the levels of, and it squashes it down into 2 dimensions. And it's a linear projection. We can talk about it, but it doesn't really matter. The point is the PCA was like the traditional way of doing this. But here I've just taken PCA and applied it to a single cell data set that has lots and lots of samples in it. It's a relatively small one, but it still has lots of samples. And we can sort of see that there's information here. Like there may be some groups of the cells that are coming out that we can kind of see, but it's a little bit noisy. And then so more recently, there's new methods that have been using statistics and borrowing some ideas from machine learning to come up with these like more nonlinear projections into lower dimensional spaces that can get, or it can capture more information about local information about, say, cell groups. And so UMAP here is on the right on the same data set. UMAP stands for Uniform Manifold Approximation Projection, I think. Hopefully I got that right. But this is a more recent, really popular way of dimensionality reduction in single cell analysis that, as you can see, just like it captures, it seems like it's capturing a little bit more information. We can see like groupings of cells that might be more informative than looking at it through a traditional PCA. Okay, so there's just 2 little flavors of places where machine learning has touched basic science and is potentially improving things. So from there, I want to just touch on one paper that I was offered in sort of, well, in this case, urine metabolomics and microbiome analyses, all basically studying drug-induced liver injury from antituberculosis drugs. And this is a cool analysis. I have the paper here. We have 2 other papers that I'm going to go through later that have, and we have 2 the authors, or one author for each of those papers, I believe, on the call. So I'll ask for them to chime in at that point. But for this one, I'll just sort of go into it, very briefly go over this paper, but just an interesting place where machine learning has touched urology, basically. So liver injury is one of the most common adverse effects of anti-TB drugs, and early identification of risk of drug-induced liver injury is critical. So here they took 4 machine learning models that were trained to predict antituberculosis drug-induced liver injury from electronic health record data, metabolomics, microbiome data, and some clinical data. And it was a small study. They had 14, say like positive patients who did suffer from drug-induced liver injury, and 30 age and matched patients who did not. But they did have high/perfect accuracy, which I have. My machine learning senses are tingling ever so slightly when I saw the results. But the point is, like, machine learning here was maybe able to then help in the future to predict, say, before someone receives an antituberculosis drug, you might be able to predict how risky is it for them to take that drug. I'll just quickly go over, like, here from the results on the left was just an analysis that they did on some differential analysis on the metabolites. So there were up-enriched and down-enriched metabolites in patient urine between the two, the drug-induced liver injury and the non-drug-induced liver injury groups that they were able to tease out. And that's just traditional statistical techniques right there. And then on the right, it's the same sort of analyses done for the microbiome data. I can't really speak to it really well. So basically, it's not exactly species that they're detecting here, but like similar type critters. And so there were like some ones that were specific to, there were 607 sequences that looked like they were specific to the non-drug-induced liver injury group, and then 189 that were specific to the actual drug-induced liver injury group. So basically, there's just like differential analysis of the microbiome between those two groups, and there were clearly significant differences. And then ultimately, they used the metabolomic and microbiome data that they collected. So some of these differentially, I want to say expressed because I work often in the bioinformatics space, but differentially enriched metabolites and microbiota, whatever, in these groups, and use the information about that, about these patients to build models to predict, are they going to have drug-induced liver injury? And they were extremely accurate. In fact, they were perfectly accurate on their test set, which is a little bit suspicious. But there's some interesting results here based on like some of the significant factors in this table up here, sorry, in this figure up here. You can see all these different features that were in the model ranked by their importance, and the ones that are the ones that are important to the model for predicting if someone is going to have drug-induced liver injury are the ones that are high magnitude. So the ones that are largely negative and largely positive, and the ones toward the zero are not as informative. But so there's some interesting biomarkers there that they can capture from using the machine learning models, and particularly from using machine learning models that have like interpretable feature weights. Basically, that's a pretty valuable outcome there. I guess like my one critique of the paper, and I won't hit on it too much further unless people have questions. My one critique would be, or I get maybe two critiques. One critique would be when they were performing their differential enrichment analyses, they were using their entire population of patients that they then used. So they used all of their data to determine what were the important features, and then separated out those patients into train and test sets to evaluate later. So in some sense, there's a little bit of information sharing between the train and test groups in the machine learning space here. That we can speak to it, but basically what I'd like to see is I would have loved to see maybe a completely held aside data set of patients. And I know it's very difficult in these types of situations, but it'd be great to have a totally blind set of patients that then they could use these determined biomarkers and see if they can predict in the unseen test set in the future. With that, the one other thing that I would want to ask, I guess, is, and I think they sort of speak to it, but they didn't speak to it really greatly in the paper, or at length in the paper, was what would be the alternatives if the risk is high. We talked about them in the previous seminars. Like the one thing that I think about when I'm working on, say, a clinically relevant problem is like, if I'm going to build a machine learning model, I want to compare to the standard of practice, and I want to know, like, if I'm building something, if my model says something, does the clinician care? And if a clinician used this model and it said, hey, this person is at high risk for drug-induced liver injury, do they care? I think they care, but what would they do otherwise? Would they pick a different drug? Would they use a different dose? All these types of questions just sort of rose in my mind, and at that point, I would want to see maybe whatever in for future work, some analysis of dose response or like individual drug response predictions instead. Anyway, I think we're still on track. So any questions on any of that stuff before we move on to more clearly clinical practice stuff?
I have a question. So
Yeah.
you were saying the critique that you offered was was it that you would have wanted to see them deal with a subset of population that they didn't know anything about and then see if the
Yeah.
model worked as opposed to knowing
Yeah.
beforehand? Okay.
Yeah. 'Cause the problem was like in my mind, and I may be mistaken, but I'm pretty confident that this is how they did this. When they were doing their differential enrichment analysis of the metabolites and the microbiota, they used the entire population of patients. So that they used all of their data to determine what were the significant features. And then when they were evaluating their model, they took some 80% of that population as like the train set and 20% as the test set, which is fine. That's normal practice. But they had already sort of used the labels on the entire data set to determine what features within this population are important. So like ideally what they would have done is taken 20% of their population ahead of time and just said like, we're not even going to touch these. And I'm going to determine what are the significant features in this 80%. And I'm going to choose those ahead of time. And then I'm going to train a model, maybe pick my favorite parameters on that 80%. And then I'm going to evaluate it on the 20%
Okay.
held aside.
So, not being a machine learning expert myself, what that sounds like to me is just a matter of a good design. I mean, when we're
Yeah.
designing experiments, even if we're not using AI, we still need to, rule out confounders and have, blinded samples and so forth. So, seems like it's just a
Yeah.
design issue, which is interesting to me that it's still relevant in AI, right? You still got to pay
Oh,
attention to
yeah.
those things. Okay.
Yeah, that's like it's one of the like I joke with people about how like it's machine learning has become so democratized and I spent such a large part of my life like learning all that stuff like getting a PhD, but now you can do all this stuff on a weekend or whatever if you're a precocious undergrad or something like that, right? And people, it's like, was all of that worth it? Did I learn anything else that I couldn't have gotten now just by waiting or something? And I think the answer is yeah. Like it's some of these details, like it's like those types of factors that you learn a little bit more about, and just thinking about the experimental side of things. So yeah, always relevant. Make sure you know you got to practice layups, I guess is the point, right? So I'm not a basketball guy. I was never good at basketball, but whatever. I'll use the analogy. Um, any other questions? Oh, I saw a chat. People want to keep me honest. Oh, great. Well,
Yeah,
you're
I just
welcome.
to say
Thank
like
you.
I totally agree and like it's so common that I see papers where there's a problem that I as a clinician don't think needs to be solved and that I wouldn't use the output of the model and it's so
Right,
critical because you can have something that performs really well and then like maybe I can't actually do anything differently.
right. Yeah. And like it can be interesting. Like maybe in this case, there's like something that can be done downstream with this, which is maybe why we popped it into the basic science side because they're like biomarkers. It's like, oh, well, maybe it doesn't change the standard of practice right now, but maybe it's like, oh, well, if you can change their microbiome in some way, maybe that affects it or something. That would be interesting to study too, very difficult. But like, yeah, absolutely. This is always the most important thing. Maybe that's another thing I learned in my PhD by working with clinicians is just like, it's all well and good to be like a machine learning cowboy or whatever and be like, oh, our accuracy is really high. But, and that's great. But at the same time, like if you actually want this to turn into something, you have to make sure it's meeting the workflow and expectations of the people who are going to use it. So yeah, 100% on board with that. And that's really common, especially in this space, I think, is to just push a paper out there and be like, whiz bang neat. And like, oh boy, this is going to change everything. But really it's not because no one can do anything about it. Any other questions that people have? Hearing none, I will charge ahead and we'll get to those other papers very soon. I promise if one of our authors on the call has to leave or something, just let me know and we'll jump right to it. But for now, I'll just talk about in clinical practice, some interesting problems that machine learning has been applied to. And there's not there's overlap between basic science and clinical practice. But In this case, let's just talk about the problem of diabetic retinopathy. This is a leading cause of blindness worldwide. I guess there aren't enough specialists to handle screening of this, and this is especially true in remote regions. But it's actually pretty scalable to capture images of the fundus. And there are really well-defined features that specialists look for when they're looking at these images that define diabetic retinopathy and different grades of it. And then and there's also just basically at this point, there's a lot of data with gold standard labels attached to them. So a lot of images with labels that say like, yeah, this is really bad, or like certain categories of diabetic retinopathy. So we have all this data. So this seems like one of these, and again, because there are well-defined visual features, this seems like it's a good place where computer vision algorithms could come into play and try to help solve this problem. So before we talk about computer vision, though, I guess like we talked a little bit about algorithms last time, but we're going to get into convolutional neural networks, which are really hot in vision, or have been at least in the last 10 years at this point. But before I do that, we'll just have to kind of like I'm going to give you like a real brief rundown of like what are artificial neural networks, which will get you an idea of like how does all modern deep learning work at this point. In really simple terms, what I can say, artificial neural networks are basically layers of mathematical transformations of data, and that's about it. So basically the point is like here, as like a brief cartoon, this is like a really, really simple artificial neural network that I've drawn here. And on the left, I have this input layer, and this could represent, each of these circles represents a value, like a numerical value. So this could be whatever, this could be pixel values, or this could be whatever, it could be like blood pressure and blood glucose. It could be all sorts, anything that's numerically measurable. Each one of these can represent one of those types of values. And then inside of an artificial neural network, we have these hidden layers. I don't know if that's even the term that people use anymore, but there are these internal layers within artificial neural networks that are then transformations of the data from the previous layer. So in this case, this hidden node, this H1, is a linear combination of x1 through x4. So it's equal to x1 times w1 plus x2 times w4, this arrow, x3 times w7, this arrow, and x4 times w10. And then that summation is then put through some sort of nonlinear or linear transformation as well. So it's the sum of that, it's that linear combination put through, in this case, it's like a sigmoid transformation. So that squashes the result into a 0 to 1 range. And the same is true for H2 and then H3. And then at some point, we might use this network to have an output, which can be a label, or it can be some other transformation of the data of interest. But basically, it could be like a true or false label, like does someone have diabetic retinopathy or something like that, right? So it could be just a binary thing. But ultimately, the point is that is also the same sort of thing. It's a linear combination of the layer of the layer behind it, and then some transformation of that linear combination. And basically, what we do with artificial neural networks is we initialize all of these weights to be effectively random values with different flavors of randomization. And then you push data through from the input layers all the way to the output layers. And at the output, you have some value that you're expecting. And through the magic of math, we can measure how wrong were you, how off were you from the expected value. And we can figure out how to back propagate the error so that we can adjust the weights to push the weights in such a way that they will get that error, they'll reduce the error, they'll push it toward the correct answer. And you do that over and over and over again with data, and ultimately you learn these weights that get from the input to your desired output by pushing it through each of these layers of mathematical transformations. And that's kind of weird and abstract, but it feels magical. But that's the idea. It's like each layer in these artificial neural networks just represents a new mathematical transformation of the information from the prior layer. And it's a little bit more complicated than that these days. Like things aren't quite as simple as this image shows. But to give you a flavor of what a convolutional neural network is doing, because it is also an artificial neural network, it's just a deep one and one that's designed for images or for anything that has like spatial locality. But so we'll talk about it in terms of images. So in this case, our layer, the weights that we care about, say this convolution, there's this box on the left, of 9 weights that I'm going to apply to, well, it's not 9 pixels in this image, but whatever, it's 9 pixels. It's basically a patch in the image. And I'm going to apply those weights to each of those pixels. And then those will come up with some like single value that will then represent one part of some transformation of that image. And I'll just successively apply that patch across the image to produce some sort of transformation. And I'll do that over and over again, and I'll do it with thousands of these types of convolutions, and there'll be different sizes and things, and ultimately I might get to ultimately just a transformation of the image, or I'll get to some label for the image. Hopefully, that gives you the idea of what a convolutional neural network is, because it's just basically using these weights and scanning across an image at little patches of the image and trying to use those transformations of the image to come up with some output. Again, it could be a transformation of the image directly, or it could be like a binary value or something like that. Is that anyone major questions or anything like that before we proceed? Okay. I'll stop there then, and we'll move on. So going back to diabetic retinopathy, I guess, which was already hinted at in that image there. Right, so there's, like as I said before, there's thousands of these labeled images available, these fundus images. There are clear visual features within the image that specialists look at. Using convolutional neural networks, there's like some review papers on this. This is one of the older like applications of successful applications of convolutional neural networks to this clinical problem. But there's like extremely accurate results from these models that are really comparable to specialists in this space. And the models are super scalable. So once you've trained a model to detect diabetic retinopathy or different grades of diabetic retinopathy within these images, as long as you give it like the same type of like a fundus image with all the expected like size and shape and whatnot, and you put it through this model, you can get an accurate result. And so this is just a great success story of how this stuff works. And then interestingly, I guess those same types of techniques are then potentially transferable to other applications, because it's really just looking for features within patches of an image. You might apply the same sort of technique to dermatology or pathology or MRIs or X-rays, and these have been used in these applications. But I think this is one of the more successful results that have come out of machine learning in clinical practice. I don't know much more about it other than that, but I guess we'll move on to one other place where AI and machine learning might be able to help is in clinical trial optimization. So as I say, we all know, I've never run a clinical trial or participated in a clinical trial, but they take a long time. They have many phases. Phase 1, 2, and 3 are just like some of the major phases, not even all of it, right? But phase 1 can take up to 2 years, and more than 2 people, but a good number of people. Phase 2 can take up to 2 years, it takes more people. Phase 3 can take up to 4 years, lots and lots of people. So lots of people are affected, lots of time is involved. So it'd be great if we could use AI and machine learning to improve this process in any way that we can. And that might be by getting to trials with more likely to be successful drugs. Like, it's a huge shame if you get all the way through a phase 3 and just determine that some drug is not going to be useful. Whoops, that's just part of the practice. But if we could improve that, or if we could improve the process of getting patients, or like reducing what do you say, like reducing the loss of patients within the trials. But basically, there's like lots of places that companies and organizations are and can use AI and machine learning. And that'll be like natural language processing to find eligible patients from the electronic health record, predicting dropout risk of patients, predicting enrollment rates at trial locations. So like optimizing your choice of trial locations, automatically monitoring for protocol adherence and anomalies, so you can reduce any sort of adverse effects earlier. And then pretrial prediction of outcomes. So like really just coming up with better drugs to test. And then post-trial analysis to determine groups that might benefit the most. So maybe you can't, maybe there's some lack in some traditional statistical techniques to determine like, I guess this is more like individualized patient care is the point. Like maybe some drugs will be more effective for some type of patient, and this one will be more effective for another type of patient. But there's all sorts of opportunities here, and lots of companies are already like capitalizing on this. But just another place where you might not think about it, but AI and machine learning do have a place. So with that, I will sort of jump to, I believe Jack Weaver is on the call, and I have a couple slides here, and I'm happy to try to speak to it. But otherwise, I invite Jack to speak to this paper, and maybe I have some questions for you, maybe not. I don't know. Find out.
Let me just prime by just a couple of introductions. So Dr. Weaver is at Cleveland Clinic, I think, but did work with Greg Tasian, who many of the people know in the CAIRIBU community. He has a funded P20 center in our CAIRIBU community on a pediatric study where they were looking at children born with spina bifida who often have urological problems, only one of which is bladder dysfunction. But Jack, if you thanks for being here. And if you want to tell us a little bit about, the clinical or importance of this study, because lots of us don't know about this aspect of pediatric urology, and then how you came up with the research question and how you decided you needed to use AI.
Yes, thanks for having me. Yeah, I worked with. I was a fellow at CHOP under Greg from 2019 to 2022, and so this work dates back to about 2019 is when Greg got funding for this and kind of started it. So our research question was, there's a lot of variability across pediatric urologists in reviewing video urodynamics. And so there was a definite need there. And AI in 2019 was still pretty, newish. And Greg had done some work on P20 already for, ultrasound analysis using AI, and had a collaborator at Penn. And there was a lot of data at CHOP, a lot of spina bifida data, and we knew this was an unmet need. So that's when the grant went into the Pennsylvania Department of Health to apply machine learning to this data to see if we can accurately predict meaningful clinical outcomes using AI, since urologists had been failing for so long. And so the the research in this area prior to this, and there's Umpire, which many of you are probably familiar with, is a study group that kind of looks at video urodynamics standardization. But prior to that, really the data is just small case reports looking at a few spina bifida patients at each location, saying, okay, this pressure is unhealthy, this pressure is healthy. But then there's so many other factors in urodynamics data in terms of, just so much. It just felt like there's a lot of data that we're leaving out in our analysis. And so that's kind of what led to this. So one question was, what was your research question? Why did you decide on AI? How did you identify and initiate a collaboration with the right data science collaborators? So, like I mentioned, Greg had collaborated with a data scientist at Penn, who was a PhD and really had background in Alzheimer's imaging and really a brilliant guy. For this work, it was the initial grant included that gentleman. But then, during the grant, CHOP developed their own computing environment and wanted us to use that. And so we actually got a data scientist from CHOP who was very green and was mainly there just to help us learn how to use the environment. And then our Penn collaborator was still going to use that. And it didn't work out, but our collaborator at CHOP, who was very green, was, very interested and actually came to some of the VUD studies and watched how they were done. And we found this just incredibly helpful because before, when we would send the data to Penn, it was, there wasn't very much transparency to it. And then we would just get, results back. And it wasn't really that helpful. There wasn't, a constant communication. Where we had this, guy who was in our team, every meeting, who had almost no stats knowledge, but luckily Greg and I are pretty good at stats. So then he was pretty much doing the AI analysis in terms of, there was a lot, there's a ton when you do radiomics and start looking at these, there's a ton of background work that there's just no way Greg or I could have done without him. In terms of I wrote down a few things here that he did. Looking at the images, character recognition software, because sometimes there's data in the image that explains what's going on. He used the pixels to get rid of any scout images that weren't necessary to the model. Just a lot of quality control of the images. Use the DICOM tags to actually link the fluoroscopic images to the time point in the tracing. So, all of that background work that there was just no way we could have done. Maybe we, so it was just a complete collaboration between the two groups. There's no way either of us would have been able to do it on our own. But it was, the best thing was that he came, was very involved, clinically, understood the problem and really understands urodynamics now. So then the paper itself, well, let me see what other questions you guys have. As a clinician scientist, what was your role in developing and executing the AI approach? So pretty much, Greg developed the clinical question, and then pre-processing the data is huge. And, you want to send, Joey's our data scientist, Joey Logan is his name. He's third author there, who, we wanted to send him really clean data. So, the nice thing about CHOP is they started recording all of their urodynamics results in a spreadsheet that is updated after every VUD study. So we had all of that data. So we were able to kind of process that data for him, make sure it was really clean before sending it to him. We requested the fluoroscopic images, the video urodynamics data. You actually have to go into the computer and convert it to text files so that you then have, just endless lists of numbers that, you can then do the neural network on. And then, so we did all of that. Obviously, we, talked Joey through kind of what our clinical question was. And then, afterwards, his models gave us risk scores pretty much. And then we just went to town with risk scores in terms of, classifying patients in high risk, low risk. And then, so our ground truth was difficult in this study because, normally, a cancer study, the ground truth is, did they develop cancer or not? Where in urodynamics, kind of what's the ground truth? And one of the main limitations of this is the reason we're doing this study is because urologists are so bad at reviewing these studies, but we needed our ground truth to be the results of urologists reviewing these studies. So we had 5 urologists, and we had a really strict program of how they reviewed the studies, and then they just rated them mild, moderate, or severe. And then we just used the risk scores that were given through all the models to come up with, AUCs and or sensitivity, specificity, and accuracy of all these. And then, these models are good because, these tables here, the ensemble model did really good at never classifying something, misclassifying something as safe that was actually severe by the experts, down in the bottom left there. So, if the experts called it severe, there was never a time where the ensemble model called it mild. So that was like a clinically relevant, important thing for us. But yeah, that's kind of the gist. I can talk more about the paper if you want, or.
So that's what Finn's going to do. So Dr. Weaver, thank you. Just want to clear up, CHOP means Children's Hospital of Philadelphia. For those that don't know, I got a question about that. And then video urodynamics are studies that doctors use to look at how a bladder is filling and other things. And it's a moving image, right? It's a video that that you look at, which, like you said, contains lots of information. So just to clear that up. So Dr. Kuusisto is now going to kind of take a deeper dive into the methods and explain some of the technical things.
Yeah, I mean, I think Jack thinks you did a great job, covering all that. I had maybe even a couple questions that I would want to ask you, but just to sort of point out to the groups. Just to point out to the crowd, because I was sort of talking about convolutional neural networks in part because of this paper, right? You used CNNs applied both to the video, which makes perfect sense, and then also applied to these time series data about the urodynamics data, which was interesting in part because it's not always obvious to people, I think, but maybe to machine learning people it is, but like CNNs are so classically used for images because you're looking at patches of images, because patches of images, like local regions of images, have relationships, and there's like importance between the pixels that are next to each other, right? Like a pixel in the top left corner is probably not going to gain, you won't learn much about the image by looking at the pixel in the top left corner and in the bottom right corner, right? Like the pixels that are in a local cluster, there's like there's a spatial locality, there's local information there. And the same is true of time series, right? It's just that it's temporal locality, right? So in chunks of time, there's more information to be shared between adjacent time points than there are time points that are far apart. So that's why you can use convolutional neural networks for these types of data. And I thought that was really interesting that you were able to combine those things, and that's really cool. Also wanted to comment, great to hear about, like, I think this sort of speaks to the comment from earlier about really working closely with, data scientists working really closely with the domain expert, basically, right? If you want to make something that's relevant, you really need to work with domain experts, because and you have to be able to collaborate. Like, it takes time, but you have to become conversant in the application area. And just that's great to hear that you had someone who was willing to like spend the time and like sit in on meetings and stuff like that, because it's so important to actually get anything of relevance. Basically, it's just to understand the problem and really understand the customer, basically. Right. So I think there was one question I had for you that maybe you can speak to. It sounded to me when I was reading the paper, the expert reviews, you commented on how like one of the big problems is inter-rater variability. So like it's already hard to get like expert reviews and then take a majority class from that the panel of experts. But also, like, when the experts were doing their reviews, it sounded like maybe they were looking at a little bit more information than a typical clinician would look at when they were making that like in a standard setting, like when they were making their decision. Is that true, or is that inaccurate in this case?
It's tough to say. Yeah, it's tough to say because these studies were kind of in a bubble where normally a urologist will review the patient's past medical history, kind of prior studies that they've had. We'll go back and look at those old studies, where these were kind of in a vacuum, these studies, and they had no idea. The reviewers had no idea any. It was just the standalone study with the fluoroscopic images with detailed analysis that the prior urologist had, described the study, in terms of some key points, but they didn't know anything about the patient or their prior studies. So it was very artificial.
Interesting. Yeah, but I guess what I should have asked maybe is also then um, like, do you think that the labels would be as close to the standard of practice as you could get, or would they be an even higher gold standard, or what do you think? How would they compare?
Yeah, our hope was a higher gold standard. We wanted to really lay these reviews on a tee for them to really knock them out of the park. We really wanted to make sure that, and we threw out studies that, weren't of good quality, and we really wanted good studies, to be really clear to the reviewer, what the tracing looked like, and kind of what was extracted at the time of the tracing, in terms of, because sometimes just being there during the study is helpful. So we wanted them to kind of have that information as well, just to make sure that, because honestly, we don't, I know how you mentioned earlier, you want to compare your model to the gold standard of like current clinical practice, but the current clinical practice is so bad that we don't even need to really, necessarily do that. We wanted to help prop them up so that they could give us a good ground truth.
Yeah, and then I guess like to that point, then, like, just the same question I would have asked to the previous one, that what, as a clinician, if you were if you had this model and you were evaluating a patient, what would you do with the result of this model? If you like, is this at a point where you could deploy this, and like some clinician who's practicing in this space would look at the result of the model and it would be helpful in some way? And if so, like how would that help?
Yeah, this model, these models particularly, I think they could help in clinic as just, I think obviously the urologist still needs to review the analysis themselves and, the study themselves and come up with their own. But, if they review it and then they look at our model and our model agreed, that gives them a little more confidence in an area where they're otherwise, a lot of, a lot of variability. So that's helpful. And then also, if they say it's severe and it's mild, maybe that helps them, rethink and maybe ask one of their colleagues, take a look at this one. I'm having issues with it. And then, another paper this was published in 2023. We just had our AIM-2 of this study accepted a Journal of Urology that's going to be published in the next few months, where the outcome was incident hydronephrosis. So, which is a surrogate marker for CKD, which is, ultimately what we care about in these spina bifida patients is, are they having renal damage? So the model actually predicts which patients will go on to develop hydronephrosis. So, if they have if that model performs well and they have that in clinic, then they, know immediately, this patient might develop hydronephrosis, might develop renal damage. Let's increase their anticholinergics. Let's, start overnight catheter drainage. Let's do all these things to help prevent that from happening well before it actually would have showed up on the ultrasound.
Yeah, no, that's great. And that's sort of what I was getting at, because I expected this would be the type of, just going back to your original comment, like I would suspect it would be like a confidence booster or reducer so that you would get a second opinion or something like that.
Yeah.
But then also like just I guess we should move on, but I'll just one last comment or one question I'll have for you. One last comment is there's like traditionally deep learning models like convolutional neural networks are there's like sort of this balance between explainability and complexity. And complexity is good because you can increase accuracy potentially, but it's counterbalanced by lack of explainability. But I wonder also if like there would be future work for then exploring, there's like new methods for making some of these models more explainable or something like that, if for some reason, like maybe it like highlighted a region of the image rather than giving you some discrete output. If it was like, oh, hey, clinician, you should look at this area, when like I was focusing on this when I came up with my, not to anthropomorphize the model, but like, oh, the model focused on this when coming up with its results. So like maybe you should look there a little more carefully. And I assume that's an interest area. But
Yeah, I mean, that's a great goal. I haven't really even thought, I mean, we tried to kind of address that a little bit with our heat maps in the paper, but yeah, I mean, absolutely. If we could, every time we publish this, they're like, so what is the, because this urologists want to trace it back to what they know. So they want to say, okay, so what's the detussural leak point pressure that the model said? I'm like, well, it doesn't give that. It gives us a risk score. But if we could somehow trace it back to see kind of what regions, yeah. I don't know how to do that. I don't know if Joey knows how to do that, but that's a really.
I mean, these are like active areas of research. So like this it's just something that occurred to me as it's like that's just great though. This is cool stuff, and glad to hear that it's like does seem like it's like addressing an actual need. So really cool.
Thanks.
Thanks, Jack. I just want to ask, is Dr. Kuusisto on the call? I don't think so. I don't see his name.
In that
If not,
case, I can speak to it, but
great, go for it.
Okay. So then the last paper, I won't bother with the comments there. I'll let someone else summarize comments for me if need be. But last paper, I'll just quickly summarize since we're running out of time here, is this machine learning models predict kidney stone recurrence using 24-hour, whatever, just predicting kidney stone recurrence. But basically, kidney stones affect 1 in 11 Americans, and recurrence is really high. So 35 to 50% of patients that have an initial kidney stone will have another one. And this results in nearly $10 billion in annual expenses. So, I don't know, like half of NASA's budget or something like that every year. Yeah, and then current tools for predicting recurrence are really limited. And then here they compare 3 different machine learning models for predicting kidney stone recurrence from demographic, clinical, and lab data. And I guess like I had a couple of questions that I would have asked, but just whoops. Go here, like ultimately I just have the results that there aren't a lot of like super hot figures to look at, but I guess the summary was I guess maybe there's like one major takeaway that I got from this that I can comment on more specifically, but basically just that they use these multiple different models, two of which I would call more like machine learning models, and then two of which I would just call really traditional statistical models, like the logistic regression and lasso. And it was kind of, I guess, reassuring to see that they had such good results with the more traditional statistical models, because I think, and I guess I'll say, like, so these are ROC curves here, and the AUC was, above 50%. So they're like getting higher than random guessing for sure. I don't know how that compares to clinical practice, but it suggests that there's some information there. And they pointed out that some of the models, which I'll go back, sorry, I'm rambling, like my previous comment, that these more traditional statistical models were the ones that did better. And that's nice because these two in particular are often really easy to interpret, like what features were the most important and such. So that's nice. You can gather information that might allow you to do some sort of knowledge discovery. And they did confirm that some of the features that seemed to be more important in the logistic regression and lasso models were the ones that they would expect clinically to pop up. Yeah, I guess, like, to that point, like, I'd just like to, if one of the authors were here, I'd ask more about like how this would pan out in clinical practice, but interesting stuff. Yeah, maybe I'll leave it at that.